MSG? Fine by me. It's just a salt of glutamate. So why haters gotta hate? So glutamate is an amino acid letter, and it's the one that's responsible for the umami or savory taste. It's a total myth that MSG causes weird symptoms, as we'll get into, but there is a lot of true stuff about glutamate, including its ability to form like salt bridges between different regions of proteins, and its ability to do like this transamination. So basically, take the amino group from other amino acids and safely transport it to like the liver and stuff where it can get safely removed. So lots of true cool stuff about glutamate. Let's start off with breaking that myth.
So let's start by saying that chemicals are not all bad. In fact, if it weren't for chemicals, we wouldn't be alive. If it weren't for chemicals, like we wouldn't exist. We are made of chemicals. Everything is chemicals. And there's only certain certain chemicals definitely can be dangerous. But the idea of chemicals in general being bad is just a total myth. And it's a total way that companies can use to kind of prey on our fear of like these weird terms. So scientists often use a lot of like weird terms or like scientific jargon. And so part of my thing with my bumbling biochemist is to try to make this stuff more accessible. And so let's talk about what these terminology regarding MSG is actually talking about. So MSG, or monosodium glutamate. Well, if you talk, so basically that's just the scientific name for this. We're just naming what's in this thing. And so a lot of times as scientists, we have like common terms for things that we use. So for table salt, for example, that's an sodium chloride. So if we called that monosodium chloride, that would sound a lot scarier than just salt, right? How about water? If we said like dihydrogen monoxide, that sounds a lot scarier than water. And so by naming things like by their proper scientific name, it allows scientists to more easily know what's inside of something and to be able to compare things and that sort of thing. But it can also make it harder to access, and it can make things kind of sound scary if you don't know what's being talked about. So, and just like you might see on like labels or something, if something, you might have heard like if something has a long name you can't pronounce, then it must be bad for you. That's totally not true. It just like some things which we don't have the common, we don't use the common name for, or if we make a version of it in the lab, that's basically the same thing, but maybe it's more stable or something, or maybe it's the exact same thing, we just give it its scientific name, then it can sound really scary. And so don't let people prey on your fears of that sort of thing in order to sell you products that they claim are like chemical free and stuff, because really that doesn't exist. And a lot of the time when you have like a natural product, it's really can be like worse for you than a lab made version of the same thing. And so, yeah, like with food coloring and stuff, like sometimes the natural flavorings are made from like insects stuff instead of like if you just made it in the lab. But anyway, I'm not going to get too far into that. Let's talk about MSG. So MSG is monosodium glutamate. And so the name, as the name suggests, it has one monosodium molecule, sodium. We call this the cation because it's positively charged. One glutamate, this is our anion, so it's negatively charged. You have a positive and a negative charge cancel out to make a neutral molecule. And when we have this happen, we call it a salt. So this is a monosodium glutamate, is the salt of glutamate, and glutamate is just one of our amino acids, and so it is one of our protein letters. So there are 20 different common ones of these, and basically the genes, so the instructions for making one of these proteins is going to tell your cells, so the protein making machinery called the ribosome, it's going to tell them what order to link these up in. Then they're going to get linked up, and they're going to link up through this generic backbone part that they have with this amino group and this carboxyl group, and they can link together through these peptide bonds. And then they each have this unique side chain or R group that sticks off, and this is going to give them unique properties. So as we've gone through these 20 days of amino acids, we're almost to the end, and we've seen that these amine side chains have a lot of different properties, and that can influence things. And so when we're talking about glutamate, it's one of what we call the acidic amino acids. So we'll get more into this, but basically it can act as an acid. It's in its neutral form, it has this carboxylic acid group. It can act as an acid, donate a proton, and then it becomes this negatively charged carboxylic group. And so because it has this negative charge, then if we put it with something positive, we can use a counter ion, so something that has the opposite charge. So it has a negative charge, so it's anionic. We put it with something that has a positive charge, so a cation. In the case of MSG, our cation is going to be sodium. You put those together and you get a neutral compound. And when we have one of these, we call it a salt. And so monosodium glutamate is just a salt of glutamate. So imagine if you tried to stick a bunch of glutamate molecules together, because they're all like negatively charged, they're not really going to want to hang out with one another. So you add this counter ion to help stabilize the situation and allow you to make this flavoring that you can then use and give it that savory umami taste. And so it's a common food additive. And it's just like a total myth that it causes all this weird stuff. So the myth about MSG started in 1968. So this is around the time when like Rachel Carson's Silent Spring had come out and people were starting to really worry about chemical additives in food. And then the New England Journal of Medicine, which is like this well-respected journal, it published this article from it published this letter. It wasn't even an article, it was just a letter, like letter to the editor type of thing. And there was this doctor who had eaten in that Chinese restaurant, experienced some weird symptoms, and suggested that the cause might be MSG. And so then that was enough, the suggestion was enough that other people started writing in. And then newspapers started publishing attention-drawing articles, and then the whole myth took off. And it was aided by what's sociologists call like cultural construction. So basically drawing on negative stereotypes of Asian countries as like dirty. And so this led to the term Chinese restaurant syndrome. But the truth is that MSG is in a lot of things, like way more than just Chinese food. It's in canned food, soups, processed meats, etc. And there's been a bunch of studies and that people like who think that they have sensitivity to MSG, they don't report symptoms if they don't think that they're eating MSG. So some people might actually have some sort of allergy to it, but what most people experience is something called the nocebo effect, which is basically like the opposite of the placebo effect. So you experience real symptoms based off of a false premise, and then false premise beliefs are hard to break. So much more on this in my blog, and even more this great article from 538 by Anna Maria Berry-Juster. And so I encourage you to check those out. So let's try to break down these concepts even more and really de-jargon stuff, figure out where these charges are coming from, and also talk about glutamate in general and all the cool functions that it can play in our body, in addition to just like making food taste good. Sodium is just an element, as is nitrogen, hydrogen, carbon, oxygen. All these are just different elements. And so like all these are just all like the different things if you that you see, like when you see a periodic table, these are just referring to these different elements. And basically all of these elements have the same general makeup. They're made up of these smaller things called subatomic particles. So they have these positively charged protons and these neutral neutrons that hang out in this dense central core, and then whizzing around them is this cloud of negatively charged electrons. So different elements have like different numbers of protons, and the number of protons is fixed. And so, for example, carbon is always going to have 6 protons, nitrogen is always going to have 7, and oxygen is always going to have 8, hydrogen is always going to have 1. But the number of electrons can change, and so they can give and take electrons, and they can share electrons, and they can share electrons to form what we call covalent bonds. So for reasons outside of the scope of this video, but more in other videos, these want to have like a full valence shell, so the full shell of these further out electrons. So this is just a simplified view of things. These electrons are really in like these like orbitals and stuff, which can It's a lot more complicated. But basically, we can understand a lot if we think about it in these like shell models. And they're wanting to get a full outer shell. And so they're going to form bonds in order to do so, or they can give and take protons in order to do so. So when you have a covalent bond, basically here they're sharing pairs of electrons, and this is going to allow them to form bonds. And now they're each fulfilling their shells. So hydrogen wants 2 and oxygen wants 8. So they join together and now they're both fulfilled. And we call this a covalent bond. And in these bonds, they're actually like sharing electrons. They're like merging those electron clouds kind of. These are the strong types of bonds that we draw as like solid lines and that sort of thing. These are the bonds that are linking together all of the different molecules of like glutamate. This is held together by covalent bonds. All of these lines are covalent bonds. But that's not the only thing that's not the only way that atoms can get this like full shell. And so another way is by like giving and taking electrons. So if we look at sodium and chloride, so sodium chloride is basically table salt. And what's happening here is we get this thing called an ionic bond. So basically sodium, if it loses a proton, then it has a full shell underneath it. Sorry, if it loses an electron, it has a full shell underneath it. And so that'll make it happy. Chlorine, if it has it only needs 1 electron to fulfill its shell. So if sodium donates an electron, gives it to chlorine. Now chlorine has a full shell, sodium has a full shell, and they're happy. But now they're also charged. And so atoms don't really like being charged. And if they are charged, they want to hang out with something that's oppositely charged. So you get this full charge attraction. But the electrons were given and taken, they weren't shared. So they're not like merging up those electron clouds or anything. They're just hanging out close to each other because you have these opposite charges attracting. And so this is a strong attraction, but it's not like a true bond. And so this means that we can like, if we put it into water, if we put it like dissolve it, we dissociate it so we can like split these apart. And these bonds these like quote unquote bonds can really, they're reversible. You can make and break them and that sort of thing because they're not really bonds. They're just hanging out together because they're charged. So because these are involving like full charge attractions, they're stronger than these like partial charge attractions that we've seen a lot when we have things that basically the molecules, they just don't share their electrons fairly. So even though they're neutral overall, we get this thing called polarity where there's parts that are more partly negative, parts that are partly positive. And so these can form these attractions. We saw a great example of this yesterday when we were looking at hydrogen bonding. And so these partial charge attractions are really, really important in biochemistry. But we can also, but these are weaker than these like full quote unquote ionic bonds, which involve these like full charges. But neither of those are as strong as covalent bonds where you're actually you're sharing pairs of electrons. So you're like merging your electron clouds. So you might sometimes see glutamate referred to as glutamic acid. And so this is because it can exist in both a protonated state and a deprotonated state. And so when it's in its protonated state, so we call these like H pluses, these are protons. When it's in this protonated state, we call it glutamic acid, and when it's deprotonated, we call it glutamate. Either case, we can abbreviate it GLU or E. And often we just kind of use glutamic acid or glutamate interchangeably because it gets kind of confusing because they can interconvert between one another because this is all just like pH dependent. So pH is a measure of how many free protons are around. Glutamate is one of 2 acidic amino acids. And so we refer to these as acidic because in their neutral form, they can act as an acid. So an acid is something that donates a proton and a base is something that accepts a proton. So in their neutral form, aspartic acid and glutamic acid can act as acids and donate a proton. And so we call these acidic. In the body, we're always way above their pKa, so we're almost always going to find them in their deprotonated state. And so you can think about if there's a lot of free protons, then it's more likely to be protonated. but there's only a lot of free protons if you're at like a really acidic pH. So this is value, and so it's only going to be protonated in that glutamic acid state in that neutral form if you're at a really low pH. So there's this value called the pKa, which is the value at which half of it is going to be protonated and half of it is going to be deprotonated. And so below the pKa, you're going to have more protons available. And so you're going to be more, the protonated state is going to dominate. And above the pKa, the deprotonated state is going to dominate because you have fewer protons. So you can compare the pH to the pKa. So the pKa is like a measure of an individual acid strength, and the pH is a measure of like overall proton availability. So when we look at the glutamic acid, so this is the form that the neutral form is going to act as an acid and donate a proton. It is a pKa of around 4.3. And then in our bodies, the pH is around like 7.4 or so. So we're about 3 pH units above the pKa. What we have in here is that we're going to see that only about 0.1% of it is going to be protonated. So we're going to have a vast majority of it be in the deprotonated state. And this deprotonated state is going to be this like glutamate state. And so where it has this negative charge. So this glutamate is the conjugate base of glutamic acid, which is the acid form. So when it's in its deprotonated form, which is the form that we normally find it in, it's actually able to act as a base. So it's able to take a proton and instead of acting as an acid, because it's already acted as an acid, it's already given up a proton. And so now we have its conjugate base, and this conjugate base form is glutamate. And so similarly, if we look at aspartate, we see that it has this carboxylic group that can act as a base as well. So this is the conjugate base of aspartate, which is the conjugate acid. So I say that these can act as a base, but they're not that likely to do so because they have this low pKa. So they much prefer to give up this proton than to take the proton. And so it's hard to get them to take a proton. And this is because you have this, basically you have this like resonance stabilization in this carboxylic group. So because the flip side of every acid is what we call its conjugate base. So once it donates a proton, it can accept a proton. Once it accepts a proton, it can donate the proton. So these protons can come and go. And so we can talk about a single molecule is acting as both an acid and a conjugate base. And so basically you have this charge is actually going to be delocalized. It's going to be shared in this group, and that's going to make it really, really happy. So it's happy in this state. And so it's a lot less likely to act as a base and take a proton than when we're looking at things like a deprotonated hydroxyl group where you don't have the resonance. There are 3 that we call basic amino acids. And so basic means that you can accept a proton. And the basic refers to the neutral state. So in their neutral state, they can act as a base and accept a proton. And this is going to make them positively charged. So with the acids, we are going from neutral to negatively charged if we acted as an acid. With the lysine and arginine and histidine, if they were to act as an acid, so then they would go from positive to neutral. So we instead of talking about them acting as a base and accepting a proton, because we talk about pKa values, which is the value of the acid donating a proton, in that case, we're going to be talking about the deprotonation. But if we were to talk about like acting as a base, then that would be going from neutral to positively charged. And we can see that these have these really high pKa. So although we talk about those amino acids, those 3 blue ones as basic amino acids, what we're actually talking about with the pKa is the strength of their conjugate acid. And basically, if you have a strong conjugate acid, if you have a weak a stronger base, you're going to have a weaker conjugate acid. So basically, if you're better at taking a proton, then you're less likely to want to give that proton up, right? And so if you have a conjugate base, if you have a stronger base, it's going to have a weaker conjugate acid. So if something is a stronger base, we'd expect it to be more protonated, right? And so we need our pKa to be further to be higher, so that we're on the left of the pKa, so that we're at a lower pH than the pKa. And so this is why with a stronger base, we're going to see a higher pKa, and with a stronger acid, we're going to see a lower pKa. And so when we have a stronger acid, we're normally going to be on the right side, so we're going to be more basic, then we're going to be at a higher pH than the pKa, and thus we're going to be in the deprotonated state. So when we talk about acidic amino acids, we're talking about aspartic acid and glutamic acid, which deprotonate to form aspartate and glutamate. And we're always on this like right side of, we're always more at a higher pH than their pKa, so we're going to see them deprotonated. With the lysine and the arginine, we're on the lower than their pKa, and so we're going to see them protonated. With histidine, it's a bit, we're closer to the pH, we're closer to its pKa, so it's predominantly going to be in the deprotonated state, but there's also going to be some of the protonated, and we can go back and forth between the two, and it's really important for like enzymes and that sort of thing to function and help speed up reactions. And the reason why I'm taking all of this time to tell you about these like acids and bases and stuff is because in addition to helping make reactions go, they can actually do cool stuff. And so they can form what we call salt bridges. So we saw how we could have a salt when we had like a MonoSodium Glutamate or whatever. That would be like a salt. We can also refer to like salt bridges and proteins. And what these are is just like, this is just another term for like an ionic bond. And it's more commonly used when we're talking about amino acids. So like an acidic and a basic amino acid of a protein, forming one of these ionic bonds. And this is going to help contribute to what we call the tertiary structure and the quaternary structure of proteins. So basically they're 3D shapes. So these ionic bonding is going to play less of a role than you might think in like protein structure and that sort of thing. The hydrogen bonds and stuff, even hydrophobic interactions, all sorts of these other interactions that are like are actually going to play a really predominant role. And part of the reason is that these amino acids, these like basic and acidic amino acids, they're competing, like your protein's like in like this watery solution. And so water can actually, basically, because water has these like partially charged regions, it can kind of like hide these charges. And so the effects aren't as big as you might think. And so you can get salt bridges and proteins and stuff. It's not as big of a deal as you might think, but it's easier. It's one of the easier things to explain when you're talking about like why regions of proteins might want to stick to one another. So it's a good kind of like starting point. So now that we've gotten this basic, the basics of all of this down, let's talk more about what glutamate can do in all the cool stuff glutamate can do in our bodies. And so of course, one of the key functions is to make proteins, but that's way not the end of the story. So glutamate is actually it's like our major amino acid. It's like the most common amino acid in our bodies, and because it's this way because it's the major nitrogen donor in metabolism. And so let me start by saying that, so if you look up glutamate and stuff, a lot of what you see is about glutamate's role as a neurotransmitter. So basically it can help it can help like neurons, so like brain cells, connect with one another. And so basically glutamate can get released by one neuron and then like taken in by another neuron, and it helps send signals that allows them to communicate. Glutamate can also be converted into GABA, so gamma butyric acid, which is like an inhibitory signal. So you can have glutamate, which excites things, and then GABA, which inhibits things, and so you can make this balance of the glutamate to the GABA. And there's all sorts of this brain stuff, but I'm not a neuroscientist, so I'm not going to try to get into that. Plus, there's like way more stuff about that part of that sort of information about glutamate than there is about the other cool stuff it can do, at least if you just like initially Google it. And so I want to talk about that other cool stuff. And this other cool stuff is largely going to involve its role as a major nitrogen donor in metabolism. And so metabolism is just like the making and breaking of molecules. So we can talk about catabolism, which is breaking down molecules, anabolism, which is building up molecules. And so glutamate is going to act in both of those types of situations as this nitrogen donor. So why glutamate? Well, part of it's kind of like you can think of, well, our bodies are kind of lazy. Like we don't want to have to make extra work if we don't have to. So with any amino acid, if you want to break it down, if you want to catabolize it, you're going to have to remove the amino group. So with from the alpha amino group. And so when we talk about amino acids, we refer to this amino group as the alpha amino group because we refer to this central carbon as the alpha carbon. So C alpha. And so then they're each going to have like a unique side chain, and this side chain in some cases has a nitrogen group, but they're all going to have this nitrogen group in this backbone. And why I talk about this, why I mention this is because your body needs to like tightly control the release of nitrogen. Because like if you have too much ammonia, that can be like toxic to your body. And so often what happens is this thing called transamination. So we take the amino group from an amino acid, stick it onto alpha ketoglutarate to make glutamate. So alpha ketoglutarate, it's this component of this thing called the tricarboxylic acid cycle, sometimes called the Krebs cycle or the citric acid cycle. So this is the alpha ketoglutarate we're talking about. And so it's just key metabolic intermediate of this cycle that can be used to convert, to produce energy, to convert things into forms that can then be used to make other things, all sorts of cool stuff. But basically, so we have this alpha ketoglutarate, this 2-oxoglutarate is going to serve as like a key hub in a lot of ways. And that alpha ketoglutarate is just one step away from becoming glutamate. It just needs to switch out this carbonyl, so the C double bond O, for an amino group, so a nitrogen hydrogen group. And so basically we can take the amino group from any amino acid. Well, not any. So basically there's a transaminase. So some of them can do like from multiple amino acids. But there's at least one for everything except lysine and threonine. You can transfer to alpha ketoglutarate to make glutamate. This way, you can take any amino acid, transfer the amino group to alpha ketoglutarate and make glutamate. And so now all you have to do is worry about getting glutamate, the nitrogen group from the glutamate release safely. So here, basically, we're saving work because we only need to focus on getting it off of this one safely. So of course, yeah, so we need to spend some energy to make different enzymes. So some of them transaminases can act on like multiple ones. Some of them are specific, but in any case, it's less important that we're not trying to safely release the ammonia or anything. We're just trying to transfer the group so that we can then focus on safely removing it from glutamate. And so we can safely remove it by kind of localizing where we're going to be doing this oxidative deamination, where we're going to be removing the group. And so we can, for example, remove it in the liver. And then if we remove it in the liver, then it can be used to make urea. And so the liver is able to do this thing called the urea cycle. And so we can then take that ammonia and make urea from it, which can be safely excreted. You can also have it happen in the kidneys. And in this case, the ammonia is released as ammonia, but it can then serve as a counter ion to promote the excretion of acids and help maintain the pH balance. And so remember that just as we saw with our salts, where you can have like a positive and a negative charge. So if you have something that's acidic, something that's acted as an acid, and is now negatively charged, now you can counter ion it up with a sodium, And you can this way help promote the excretion of acids. And so it can play this important role. But in either of these cases, what you're doing is you're controlling where you're releasing the amino group from the glutamate. And that amino group could have come from any amino acid. And so this way, it makes sense to go through this work of transferring the amino group from anything to glutamates. And so that glutamates can then be used as this nitrogen donor. But that's if you want to just like release the amino group, like get rid of it. You can also, a lot of times you're going to want to like transfer that amino group. For example, we saw the other day how we could use glutamate to make aspartate. So take the amino group from glutamate, stick it onto oxaloacetate to make aspartate. But then we also saw a reaction more like what we just looked at, where we took aspartate and we stuck its amino group onto alpha ketoglutarate to make glutamate. And so this brings up a key point, is that these are enzymes. So these are reaction speeder uppers. So basically they're lowering the activation barrier to make a reaction happen, but this is going to speed up the reaction in both directions. So it makes it easier for a reaction to occur, but the reaction can occur, it can kind of go in either direction. And so enzymes can make this reaction happen in either direction, and from which direction it goes is going to be regulated in large part by like how much of these starting products you have or the ending product. So if you have more of the starting stuff, it's going to help push the reaction in this direction. If you have more of this stuff, it's going to push the reaction in this direction, and that sort of thing. So basically the enzyme, it'll take it, make it easier to get to the transition point, at which point it can fall either direction. But if your products are at a lot lower energy, so if your products are a lot more stable than your reactants, it's going to still have a higher barrier. It's to actually going back the other direction. And so you're going to end up having a reaction that's dominated in one direction or another, in addition to having just the effects of like getting to the activation state from the reactants. And one way that we can make the reactions more favorable in like one direction is by coupling it to something that makes it really, the products a lot more happy than the reactants. And so that it's really hard to go backwards. And the way that we can do this is by using like ATP, which is kind of like energy money. And so basically you can think of these negatively charged phosphate groups as kind of being kind of like a clamp spring, like so holding them together, kind of like holding in a lot of energy because you have all of these negative charges next to each other. If you break them, you're releasing energy. And this phosphate group is going to be really not going to just like hop back on next to all these other negatively charged phosphates. So if you couple something to the release of energy through ATP, then this can help make the reaction go in one direction as opposed to going backwards. And we see this if we look at the making and breaking of glutamine. So glutamine, which we looked at yesterday, it has this amino group instead. It's like amide group instead of this carboxyl group. And so basically you need to swap this out for an amine, the nitrogen group. And so in order to do this, we're going to couple it to the use of ATP. And we're actually going to get like stuck on ATP as an intermediate. And this is going to allow us to make glutamine. And you can see that this reaction would be very unlikely to go in the backwards direction because that would involve this like ADP having to kind of like take this phosphate group back and then like kick us off or whatever, but you don't want to stick those negative charges back together, right? And so this is going to want to go in this forward direction, and it's going to be hard to get it, like this is going to be practically irreversible. But we want to be able to make glutamate from glutamine as well. And so in order to do this, we're going to use a different enzyme, so a different reaction speeder upper. And in this case, what we're going to use is glutaminase, which is going to basically use hydrolysis to cut off this amino group to give us glutamate and ammonia. And so we can do this thing. So in our kidneys, we can basically take the glutamine, use glutaminase to release the ammonia group to get glutamate, and then use glutamate dehydrogenase to release that the second ammonia group. Because so when you have glutamine, you have two amino groups that you need to get rid of. And so when we have this happening in the kidneys, then it can be doing that pH buffering role. How about glutamate dehydrogenase? So here, this enzyme can actually go in both directions. So both of these can actually go in both directions. What's really cool about glutamate dehydrogenase, so here we're going from glutamate to alpha ketoglutarate. We're not going from glutamate to glutamine. So we're removing this, this amino group. And so this alpha amino group. This is going to use, this is like a redox reaction, so oxidation, reduction. So basically oxidation is loss of electrons, reduction is gain of electrons, much more on this in other posts. But basically, when you have glutamate, we're oxidizing it, we're forming more of these bonds to oxygen, we're losing electrons, this, at least from the carbon's perspective. And so this is going to be an oxidation reaction, and so we're going to have to reduce something in the process. And the thing that we're going to be reducing is going to be like NAD+. And then if we go the reverse direction, we have alpha ketoglutarate, we need to reduce it. So we need to oxidize something, and so we can oxidize NADPH. And so basically we've talked about NAD+ a lot, but there's also this like NADPH. And what's really cool about this glutamate dehydrogenase is that it's going to use like different versions. So it's going to use NAD+ when it wants to break down the glutamate to oxoketoglutarate. And it's going to use the NADPH if it wants to build molecules. And so basically both of these can work in the kind of the same way. But our bodies tend to use NAD+ slash NADH for the like energy for catabolism, and NADP+, NADPH for anabolism, so like building molecules. And so your cells are going to keep more of the NAD+ in its oxidized form and more of the NADPH in its reduced form. And so by having this glutamate dehydrogenase use like these different versions in either direction, then it can kind of use the version that our bodies have more in stock of. And this glutamate dehydrogenase is also going to be like allosterically regulated. So basically it's regulated by molecules that are binding to somewhere other than like the active site, so other than where this reaction is actually taking place. And they're going to be kind of like helping, like they can like change the shape of the enzyme and that sort of thing to regulate its activity. In the case of its enzyme, it can be regulated by like ATP and ADP. And so you can further control which direction you want the reaction to go in, and when you want to be making and breaking based on the cell's energy needs. So when you have high ATP, your cells are basically like, okay, we don't need to break down as much. And so the reaction is going to be favored to make more glutamate. When you have less ATP, you're going to have more ADP, so more used up ATP. And so then you're going to be like, okay, we need to make more ATP. And so you can then go in this direction, make alpha ketoglutarate, which can then go through TCA. And in the process, you're taking NAD+ and making more NADH, which can then be used in oxidative phosphorylation to give you an energy boost. Final cool note, so these transamination reactions, more on this in the lysine post, but they go through this cool thing called the shift base that's involving this PLP, which is a cofactor. So it's basically this little vitamin-like thing that the enzymes use as a helper. And there's this cool reaction mechanism you can check out in the lysine